Hello guys, welcome to BIOGURU, to your own channel. So now we will discuss about the molecular basis of inheritance. So first of all, structure of DNA. So DNA as an acidic substance present in nucleus was first identified by the Frederick Mischer in 1869. तो फ्रेडरिक मिशर ने सबसे पहले उसे न्यूक्लियस में ऑब्जर्व किया, आइडेंटिफाई किया एंड ही नेम्ड इट न्यूक्लिन बिकॉज वो न्यूक्लियस में था तो उसे नेम दिया गया न्यूक्लिन. However, due to technical limitations in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. क्योंकि अब DNA को इंटेक्ट फॉर्म में बाहर निकालना, उसे एक्सट्रैक्ट करना बहुत टफ था, इसलिए उसका जो स्ट्रक्चर था वो किसी को भी क्लियर नहीं हो पा रहा था. So it was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. And double helical structure of DNA you know very well, but I will explain in detail. So Rosalind Franklin, a student of Maurice Wilkins at King's College in London, took X-ray diffraction pictures of crystalline DNA and वो जो pictures थे उनके basis पर double helical model दिया गया. One of the hallmark of Watson and Crick proposition was base pairing between the two strands of polynucleotide chain. Base pairing A T के साथ पेयर करेगा, T A के साथ पेयर करेगा, G C के साथ and C G के साथ. However, this proposition was also based on the observation of Erwin Chargaff that for a double-stranded DNA, the ratio between adenine and thymine and that between guanine and cytosine are constant and each equals 1. क्यों? Because adenine and guanine, cytosine and thymine, प्यूरिन एंड पायरिमिडीन, तो प्यूरिन्स के साथ हमेशा पायरिमिडीन पेयर करेगा और पायरिमिडीन के साथ हमेशा प्यूरिन पेयर करेगा. Along with Wilkins, Watson and Crick were awarded Nobel Prize in 1962. And the base pairing confers a unique property to the polynucleotide chains. The chains are complementary to each other. Therefore, if the sequence of bases in one strand is known, then the sequences in other strand can be predicted because they are complementary. और अगर complementary है, अगर एक strand आपको पता है तो दूसरा strand आप predict कर सकते हैं. Also, if each strand from a DNA acts as a template for synthesis of a new strand, the two double-stranded DNA thus produced would be identical to the parental DNA molecule. अब एक स्ट्रैंड आपको पता है और दूसरा स्ट्रैंड अगर उससे फॉर्म होगा तो आप उसको प्रेडिक्ट कर सकते हो. सेम एज अगर दो डीएनए स्ट्रैंड आपके पास है और दोनों ही पेरेंटल डीएनए की तरह या टेम्पलेट डीएनए की तरह एक्ट करेंगे तो उनसे न्यू डीएनए फॉर्म होगा वो बिल्कुल आइडेंटिकल होगा because of the complementarity. Because of this, the genetic implications of the structure of DNA became very clear. So now, structure of DNA. So James Watson and Francis Crick proposed that DNA consists of two strands which are helically coiled. The double helix is right-handed and is referred to as the B form or BDNA and is the most common model. देखिए वैसे तो there are many types of DNA, ADNA, BDNA, cDNA, ddNA, fDNA, zDNA, but BDNA is very common. जो हम बुक्स में पढ़ते हैं it is actually BDNA. DNA is formed of millions of deoxyribonucleotides. The salient features of the double helix structure of DNA are as follows. It is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. There is a backbone of sugar phosphate, sugar phosphate, sugar phosphate, and bases are projected inside. And the two chains have anti-parallel polarity. This means that if one chain has the polarity 5' to 3', the other has 3' to 5'. And the bases in two strands are paired through hydrogen bonds. There are hydrogen bonds between the bases A and T, G and C. There are double hydrogen bond between adenine and thymine and adenine. And there are triple hydrogen bonds between guanine and cytosine and guanine. Adenine forms two hydrogen bonds with thymine from the opposite strand and vice versa. Similarly, guanine is bonded with cytosine with three hydrogen bonds. As a result, purine always opposite. So purine always comes opposite to a pyrimidine. This generates approximately uniform distance 20 angstrom between the two strands of the helix. The two chains are coiled in a right-handed fashion, and the pitch of the helix is 3.4 nanometer, and there are roughly 10 base pair in each turn. तो एक complete turn में total 10 base pair ही यहाँ पर आते हैं. एक टर्न का मतलब क्या हुआ? एक टर्न का मतलब हुआ 360 डिग्री, और 360 डिग्री में अगर 10 बेस पेयर होते हैं देन 360 by 10, तो हर एक बॉन्ड जो है उसमें 36 डिग्री का राइज होगा. Consequently, the distance between two successive base pairs in a helix is approximately equal to 0.35 nanometer. So, the plane of one base pairs stacks over the other in a double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. So you can see the structure, and there are some grooves, minor grooves and major grooves. देखिए जो narrow है it is minor and major is very deep. देखिए अब इसमें क्या है, जो major and minor दो अलग अलग ग्रूव्स हैं, और इन major and minor ग्रूव्स में जो आपके एंजाइम्स होते हैं वो एंकर होकर फंक्शन करेंगे. So if two purines form base pair being heterocyclic, they make DNA bulged. अगर मान लीजिए कि पढ़ा हमने कि two purines form base pairs being heterocyclic, अगर दो purines आएंगे then bulge हो जाएगा. If two pyrimidines form a base pair being monocyclic, make DNA narrower, so that a purine always pairs with a pyrimidine and vice versa. Therefore, the diameter of DNA is always constant, and a specific purine pairs with a specific pyrimidine. A के साथ T, T के साथ A, G के साथ C, and C के साथ G पेयर करेगा, because of a perfect match between hydrogen donor and acceptor site on the two bases. So, the proposition of a double helix structure for DNA and its simplicity in explaining the genetic implications became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that genetic information flows from DNA to RNA, RNA to protein. Now, formation of DNA from DNA is called replication. Formation of mRNA from DNA is called transcription, and formation of protein from mRNA is called translation. So, it is unidirectional flow of genetic information. It is called central dogma. But in retroviruses, RNA acts as a genetic material. For example, in HIV, the flow of information is in reverse direction. So it is called reverse central dogma, because there is formation of RNA. देखिए, there is formation of DNA from RNA. अब RNA से DNA फॉर्म हुआ. जनरल प्रोसेस क्या था? DNA से RNA फॉर्म होगा, इट इज कॉल्ड ट्रांसक्रिप्शन. बट RNA से DNA फॉर्म हुआ, इट इज कॉल्ड रिवर्स ट्रांसक्रिप्शन. And this process or this method is called reverse transcription. So now some MCQs. DNA double helix, the right-handed is D form, B form, Z form or X form. Yes, it is B form. DNA is formed by millions of nucleotides, nucleosides, ribonucleotides and deoxyribonucleotides. Yes, it is deoxyribonucleotides. DNA duplex has two strands with a diameter of 10 angstrom, 20 angstrom, 30 angstrom and 40 angstrom. It is 20 angstrom. Now packaging of DNA. जो DNA है वो chromosomes कैसे बनाएंगे? Because DNA and chromosome actually they are made up of simple material that is called deoxyribose nucleic acid. But DNA coil होने के बाद chromosomes form करते हैं. So now packaging. Taking the distance between two consecutive base pairs as 0.34 nanometer, the length of DNA double helix in a typical mammalian cell if calculated simply by multiplying the total number of base pair with distance between two consecutive base pair, that is 6.6 into 10 power 9 into 0.34 into 10 power minus 9 meter per base pair. Turns out to be approximately 2.2 meters. तो अगर जो डीएनए है अगर उसकी हम लेंथ मेजर करेंगे तो इट इज अबाउट 2.2 मीटर्स. अब ये 2.2 मीटर्स का डीएनए एक छोटे से सेल के न्यूक्लियस में पैक कैसे होता है? So this length is far greater than the dimension of a typical nucleus, approximately 10 power minus 6 meter. So how is such a long polymer packed in a cell? So now it is called packaging of DNA. So now first the length of eukaryotic DNA is far greater than dimension. यह भी डिस्कस किया हमने, न्यूक्लियस का साइज बहुत छोटा है, उसमें कैसे आएगा? The DNA of prokaryote and eukaryotes packed in a space which is 1000 to 1 lakh times smaller. And such a packing occurs differently. So, packing in prokaryotes. देखिए prokaryotes and eukaryotes दोनों में काफी डिफरेंस है. Prokaryotic DNA and eukaryotic DNA. Prokaryotes में प्रॉपर क्रोमोसोम्स नहीं मिलते and eukaryotes में प्रॉपर क्रोमोसोम्स बनते हैं और मिलते हैं. In prokaryotes such as E. coli, though they do not have a defined nucleus, DNA is not scattered throughout the cell. So, प्रोकैरियोट्स में भी अगर प्रॉपर न्यूक्लियस नहीं है, मेम्ब्रेन बाउंड न्यूक्लियस नहीं है, फिर भी वहां पर जो न्यूक्लियस है वो स्कैटर्ड नहीं होता. सो DNA being negatively charged is held with some proteins that have positive charge in a region termed as the nucleoid. तो वहां वो न्यूक्लियोइड फॉर्म करेंगे. The DNA in the nucleoid is organized in large loops held by the proteins. The DNA of prokaryotes is almost circular and lacks chromatin organization. तो chromatin और chromosomes का formation prokaryotes के अंदर नहीं होता. This is referred to as the ganophore. Now, DNA packing in eukaryotes. In eukaryotes, this organization is much more complex. और अगर हम बात करते हैं eukaryotes की, it is very complex. There is a set of positively charged basic proteins called histone proteins. तो histone proteins पर DNA रैप होगा, then वो न्यूक्लियोइड बनाएंगे, then solenoid, then 30 nanometer fiber, chromatin and chromatid and chromosomes. So one by one हम discuss करेंगे. Histone proteins. Histone proteins are octamer proteins because they have 8 subunits. So a protein acquires charge depending upon the abundance of amino acid resides with charge side chains. देखिए अब किसी भी प्रोटीन पर जो चार्ज है वो किसकी वजह से है? उसमें प्रेजेंट अमाइनो एसिड्स की वजह से. So DNA is negatively charged and histone proteins are positively charged, so they can be bound. Okay. So histones are rich in basic amino acid residues lysine and arginine. Both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. So, nucleosome. देखिए अब न्यूक्लियोसोम कब बनेगा? जब जो हमारे पास हिस्टोन प्रोटीन्स हैं, हिस्टोन प्रोटीन्स पर डीएनए रैप करेगा. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. तो histone octamer के ऊपर, because histone octamer is positively charged and DNA is negatively charged, वो उस पे रैप हो जाएगा. It is called nucleosome. Then a typical nucleosome contains 200 base pair of DNA helix. Nucleosome constitute the repeating unit of structure in nucleus called chromatin. The nucleosome and chromatin are seen as beads on a string. वो beads की फॉर्म में दिखाई देंगे, वो string है और उस पर छोटे छोटे beads होंगे. So when viewed under a electron microscope, so the beads on a string structure in chromatin is packaged to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form the chromosome. तो फर्दर डिवीजन होंगे, फर्दर कॉइलिंग, फर्दर पैकेजिंग होगी, देन वो क्रोमोसोम्स में कन्वर्ट होंगे. ओके, सो द पैकेजिंग ऑफ क्रोमैटिन एट हायर लेवल रिक्वायर्स एडिशनल सेट ऑफ प्रोटीन दैट कलेक्टिवली आर referred to as non-histone chromosomal proteins or NHC. In a typical nucleus, some regions of chromatin are loosely packed and stain light, and are referred to as the euchromatin. Actually, euchromatin is the active chromatin. The chromatin that is more densely packed and stains dark is called heterochromatin. It is actually inactive. So euchromatin is transcriptionally active chromatin, whereas heterochromatin is inactive. So now MCQs. Histones are rich in basic amino acids like alanine and valine, lysine and arginine, lysine and alanine, or alanine and lysine. Yes, it is lysine and arginine. A typical nucleosome consists of 100 base pair of DNA helix, 200 base pair of DNA helix, 400 base pair of DNA helix, or 300 base pair of DNA helix. Yes, it is 200 base pair of DNA helix.